Hi class, happy Monday yet again. I'm getting a jump start on the videos this week. I'm going to try to get you as much information out as possible about chapter 15.2. And hold on. Make this video while enjoying a nice glass of chocolate milk. Delicious. Alright, in our 15.2 mechanisms of evolution, that is what we are reading and learning about this week. And our objective is, how do changes in allele frequency happen? And how do those changes cause evolution in a population? And in a video I made earlier today, I discussed allele frequency. I discussed genetic equilibrium. So if you need help with that part of the 15.2 video, watch that video. Okay, today in this video, I'm going to look at how you get changes in allele frequency. And there are 4 ways that you can change the genetic equilibrium. You can change allele frequency and cause microevolution. Okay? Now, 1, natural selection. 2, genetic drift. 3, mutations. And 4, gene flow. All right, so first, with natural selection, the environment is changing the allele frequency as some alleles or versions of a trait of a gene are better suited to survive in that environment. So in this example of natural selection of a mouse population with 2 alleles, one allele is for a light brown fur, another allele is for a dark fur color. Okay, the light one gives better camouflage than the dark one. So a predator, like a fox, can and will eat more dark fur because they're easier to see. So more often, the environment will cause these dark fur mice to die, cause them to reproduce less, and these light-colored mice will survive more often and make more babies. So as generations go on, the population changes to have more of the capital A allele that gives a light brown fur and better camouflage for the environment. So natural selection, the environment is selecting one allele and making that allele more frequent in the population. Now, another example is genetic drift, and genetic drift is all chance. Okay, chance events change the allele frequency in a population. Okay, and this is pretty common in small populations. It can be caused by natural disasters. It can be caused by a small group of a population leaving a larger population. So in this picture, I have a population of ladybugs. Okay, and you have a variation in the shell color trait. And by chance, maybe wind, a hurricane, this beetle jumps on a ship by accident and gets shipped off to another island. Some chance event pushes a small population of this larger population of beetle to a new island. So now the allele frequency for that island is real different compared to the mainland or the larger population. So that population, as it grows on this island, will have an allele frequency that's different than this one. Let's look at one more example of genetic drift. So, in genetic drift, say we'll stick with the chance events. So, in my previous example, this is kind of called a founder effect, where you have a small group leaving a larger group to go to a different place. Okay. Also, the environment. Some natural disaster can happen. By chance, kills off a few. So, boom, a tree comes. Squashes those frogs. Now, all the frogs that had the allele for brown skin are gone. It had nothing to do with the environment. Heck, that might have been a mutation that was better suited for the environment. May have given them camouflage, but a tree fell on those frogs and they're all dead. So they can't pass down their genes. It has nothing to do with how good that trait was compared to the other alleles. It was just random chance. Cut them off. Okay, and this happens a lot more often in a small population. Now, if these were the only frogs, this was an endangered species, this is really likely to happen. If you have a species with only 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 frog organisms, it's really possible you can get huge changes in your allele frequency in your gene pool. Like in this case with genetic drift, where the tree falls down and crushes the 3 frogs that have the brown allele. So in your next generation, the allele frequency changes because there are no more of that brown allele. So you have a higher percentage of dark green and light green alleles in frog skin color. Genetic drift, chance changes. All right, but in those examples, there were no new alleles. Those were just changes to the gene pool of alleles that were already there. We didn't add anything new. So there are 2 ways we can get some new variation in our gene pool. One, and really the only way to add new variation on planet Earth, okay, any population, are a mutation, okay, a change in the nucleotide sequence of DNA. So we're changing the C to a T. That could be caused from something in the environment, radiation, chemicals, random chance, from errors in DNA replication. Whatever it may be, you've created a new allele when you have a mutation. You have a new sequence of nucleotides in a gene. And this is the big way that new phenotypes can be created and new alleles are added to the gene pool. Now, we're sticking with beetles. And say in our population, we just had we had one allele for beetle color, green. But a mutation comes around. By random chance, there is a mutation that creates this brown allele. It creates brown-colored beetles. Well, when that new allele is added, natural selection can work on it. So it just so happens that the birds around the beetles, in the environment around the beetles, this brown allele does a better job of camouflaging the beetles and the green ones. So the bird is going to start eating more green-colored beetles. So the allele for green starts decreasing in the population, while that mutated allele, the new allele that was created through a mutation that creates a brown color phenotype, that one starts becoming more common. So this mutation happened to be beneficial, and it got passed down. Now for us, with milk, there is a gene that makes an enzyme called lactase. And this enzyme in your small intestine breaks down lactose, a disaccharide, into its component monosaccharides, glucose and galactose. And then your body can use that sugar. The cells can use it to make ATP through cell respiration. Now, if you don't have that enzyme, you can't break down lactose. But you've got billions of bacteria in your intestines, and they'll happily take that sugar. They'll happily break it down. But in the process, they're going to create lots of gas, lots of acid. So you get those lactose intolerant symptoms of bloating and gassiness and farts and stomach pain. Now, when we're born, we all have that lactase protein. We all have the gene for lactase. Okay, it allows us to break down our mom's milk and get all the nutrition from it. But at some point during childhood, normally in humans, that gene shuts off. Okay, because you don't drink your mom's milk forever. You stop, hopefully. And at that point, that gene gets shut off because your body doesn't use it anymore. You are going to start eating grains and berries and nuts and meat. Okay, so kind of a bummer if you enjoy milk, if you enjoy ice cream, if you enjoy cheese. But some people, like me, so good. I drink milk, I can eat cheese, no problem. I don't get gassy or bloated. So in my case, I have a mutation. So in, not the lactase enzyme, but in a part of the DNA that turns on and off that lactase enzyme. I had a mutation. a point mutation. The C changed to a T. So that mutation kept my lactase gene turned on. Now, for me, that means that that lactase enzyme, it's always being made by my cells. Okay, it's always being spit out into my intestine. So when lactose comes around, I can break down that glucose. I can break down the lactose, into glucose and galactose and be happy and make energy and feel good. All right. Now, whether that stays on or off, whether it's a beneficial mutation to always have that lactase enzyme there in your intestine, it depends on your environment. Now, let's go. With me, I have Northern European ancestry. Parents or family that lived in Scotland and England and Denmark, places that can get really cold in the winter. So now if I had that mutation and I've got a dairy cow with me during the winter time, okay, I'm gonna be okay. Okay, if I couldn't find any food that day, I can milk my dairy cow, have some very calorie dense food to eat and survive. Okay? Now, if I didn't have that mutation, I couldn't drink milk. Okay? I would be on my own. I would have to forage and hunt. Okay? I'd survive, but it would just be tougher, it'd be harder. Okay? And so as a result, if I had that dairy cow, if I had that mutation, I would be able to support a lot more babies. Okay? I could support more little genes in this world, thereby passing down that mutation for lactase persistence. But if I didn't have the mutation, I'd survive and produce one son or daughter, one. But over time, as this happens over generations, I'm having a lot more babies born with that allele, that gene version for lactase persistence, for keeping that lactase enzyme on. So over time, you get more and more of the population with this lactose tolerance. Now, and when you look in Europe, you see in colder climates, there's the gene pool has a higher allele frequency for the lactase persistence mutation. So northern parts of Scotland and Ireland and Denmark and Norway and Sweden and northern France and Netherlands and northern Germany, you have a really high allele frequency for that lactase persistence allele. But where it's warmer, it's easier to grow crops. It's a little more relaxing and easy to live. The environment's not as harsh. There's no really selective pressure to have that mutation, so it doesn't show up as much. It's there, but it's not as common because the environment's not selecting for it. There's no selective pressure to make that mutation stay around. Okay, so in one spot, that mutation, that new allele, helped individuals survive and make more babies, so it shows up more in that gene pool. But in other populations where the environment's not as harsh, it's easier to grow crops and more crops, you don't see that allele show up as much. And our last example of changing the allele frequency, messing up genetic equilibrium, is gene flow. And gene flow is pretty much like it's migration, it's movement. So you have an allele, it can leave, it can emigrate, or it can enter, it can immigrate. So we'll use beetles as another example. You have a population of brown beetle lives, say, on a different island than these green beetles. And this brown beetle swims over to this island with green beetles. So now this green beetle population, it has a new allele. It has the allele frequency has changed. And if that beetle allele stays around, that brown allele stays around, natural selection could select for it. Say it does a better job of camouflaging. So over time, natural selection, the environment will select for that allele. It'll become more common and the allele frequency will change. So, in conclusion, okay, microevolution happens when the allele frequency of a gene pool changes. Okay, this can happen from a mutation being added to the gene pool. It can happen during natural selection, the environment selecting for one beneficial allele over another. You can have gene flow where new alleles enter or exit, and then you can also have genetic drift, where the allele frequency changes by some chance event. Okay, hope this helps. Keep communicating.